Our last speaker this morning is Brenton Graveley. Brent's another part of our interactive campus down here. He's at the University of Connecticut Health Center, a member of the Institute for Systems Genomics, the new campus-wide institute that's been developed as part of all the activities that are going on here, and also a member of the Genetics and Developmental Biology Department. Brent, he's going to talk about elucidating the biology of genomes. Little topic.
All right. Okay. Well, thank you very much, George, and congratulations to Ed and Charles and everyone at JAX on this very momentous occasion in the absolutely beautiful building. So as an academic scientist, it's very rare, especially in the current academic climate, to have colleagues that do the same sort of science that you do come to where you are. And so I think it's safe to say that I'm more excited than anyone in the entire state about what's going on here. So not only is JAX have this beautiful building and hiring 30 people, since the deal was brokered between JAX and UConn has decided to go all into this too, and so there's about 20 faculty that have been or are going to be hired in the area of genomics as well. So from like 2 years ago until 2 years from now, that's about 50 faculty working in genomics that are being recruited to the state. So it's really quite transformative, and I'm very excited about it. And there's been a lot of talk about these collaborations between UConn and JAX, and just to highlight that this is not really talk, since JAX has come here, I've already submitted grants or am working on them with 6 of the 14 faculty here, and also some faculty at Bar Harbor. So this is really something that's happening for real. And this is a good example of this. This is a picture of Ijun here, as well as several members of our Department of Genetics and Developmental Biology. This is Mark Lalonde, the chair of our department, and Mark was part of the delegation that drove to Bar Harbor to actually get this agreement going. So a lot of thanks goes to Mark as well. But we went to Paris earlier this year, and in addition to having scientific talks, we actually made an outline of a center grant that we're going to be submitting later this year. So we're very excited about this. So what I'd like to talk to you is about the work that we've been doing in my lab as part of the ENCODE and ModENCODE projects and some other experiments as well, and with the really idea of elucidating the biology of the genomes. And I think as Eric really nicely pointed out earlier this morning, when the human genome was sequenced, this is what we had. And our understanding of actually how to read that sequence was sort of analogous to that of an elementary school child. So our grammar was really poor, we didn't know big words, and our vocabulary was poor. So if you were confronted with something, a very huge word, you would have no idea what it means, you couldn't recognize it. And in addition, if you were given, for instance, a poem by E.E. Cummings that has no punctuation whatsoever, and the words are all mixed up and the paragraphs in different order, you wouldn't even know how to recognize it in here. So we knew how to recognize some genes, but there was a lot about the genome that we had no idea about. So several years ago, the NHGRI decided to launch this ModENCODE project, and my lab was part of this, working on the fruit fly genome. And at the time, the fruit fly genome was really one of the best annotated genomes of any organism that we had to date. And so we were really interested in interrogating the RNAs that were expressed from the genome throughout time and space. So these are just some examples of the types of samples that we've interrogated. So we interrogated many time points throughout development of the animal. We looked at many different dissected tissues from the animal, many different cell lines, and then we had many different environmental perturbations. So metals, we would cold shock them, heat shock them, put them with pesticides, give them caffeine, give them martinis. This is resveratrol, and we would infect them with viruses and whatnot. So in all of this, we've sequenced both messenger RNAs as well as total RNAs for all of these samples. There's about 500 different samples in total, and we have about 100,000-fold coverage of the entire genome. So I think we've done a fairly good job at looking at this. And so this is basically the overview of our results. So in 2008, this is when the project actually started. There were almost 15,000 known genes in Drosophila. And one thing I'd like to point out here is when we actually submitted this grant, Illumina did not sell sequencers. Okay, so this was all going to be done by microarrays. And so since then, 454 has come and gone, and Illumina is taking off, and there's obviously a new wave of sequencing technology coming out now doing these single molecules. So as many people have already said throughout yesterday and today, we don't even know where we're going yet because we don't know what's there. So it's a very exciting period of time. But we identified over 2,000 new genes in doing this, which was kind of remarkable considering how well annotated this genome was. So one of the reasons that we missed these before was really due to two things. So first of all, this is one example of a new gene. So these are 30 different time points throughout development. This is RNA sequence data. So this particular gene down here is the new one. What you can see is it's really only expressed in one of these 30 different time points. So if you didn't have the right exact sample, you wouldn't ever see it. And then the other thing is this track right here is the conservation across insects. And many computer programs that predict genes use conservation to identify them. And this gene is completely unconserved. So it's really a new gene that's really specific to the melanogaster species. So almost all of these new genes that we identified are very temporally or spatially restricted in their expression patterns. And they have features such as not being conserved that all the computer programs would miss. And then there were other things like this. So this is looking at these environmental perturbations. There were many of the genes that we identified that were only expressed in one of the environmental perturbations. So these are environment-induced genes. One of the interesting things is one of the third most upregulated gene is a non-coding RNA. And so in looking at these non-coding RNAs, we identified about 3,800 genes that have ORFs less than 100 amino acids. 795 of these seem to encode proteins that are 20 amino acids or more. And so we have about 1,800 candidate non-coding RNAs in Drosophila. That's just one example of them here. But one of the interesting things is we can look at the database of all the mutations that have been made in Drosophila and look for any phenotypes that might exist in mutations that overlap these non-coding RNAs. And it turns out that every single case of a mutation that overlapped one of these new non-coding RNAs, they had a phenotype. Okay, so we think these non-coding RNAs are doing something that's important. So that's a lot of new genes we discovered, but really the biggest discovery was the number of known transcripts. So we went from about 25,000 transcripts to over 300,000 different transcripts. And so this is really through the process of alternative splicing. But if we look at this, most of the genes in the genome make only a few transcripts, but there's actually about 100 genes or so that make up the majority of the transcripts that are expressed from the entire animal. So there are some genes that make thousands and thousands of isoforms, and I'll tell you a little bit about some of those. So 47 encode over 1,000 transcripts each. Half of these are only expressed in the embryo, 27 are only expressed in the nervous system. And then of these, about half have these extensions of the 3' UTR that are very, very long in the nervous system. 44% of these are edited, and 21% of those have both editing and these very long UTR extensions. So if we want to talk about lots of isoforms, this is a gene that my lab has worked on for many years called Dscam for the Down syndrome cell adhesion molecule. This gene has 115 exons, and 95 of them are alternatively spliced. So there's this exon 4, 6, and 9 clusters have 12, 48, and 33 exons each, and then this one has a small number of 2. And the exons within each of these clusters are spliced in a mutually exclusive manner. So every time you make a messenger RNA, you use one of the exons from each of these clusters. And so it's theoretically possible to generate about 38,000 different isoforms from this single gene. And actually some sequencing experiments that my lab and other labs have done have verified that nearly all of these are actually detectable in the animal. And the splicing of exons 4, 6, and 9 changed the sequence of immunoglobulin repeats in the extracellular domain. Exon 17 changes the transmembrane domain. And some really beautiful work from Larry Zapersky's lab has shown that depending on which isoforms of this gene are expressed in neurons determines the wiring pattern of those neurons. And this gene also plays an interesting role in the immune system in the fly as well. So one thing that we've been interested in doing is developing basically a map of the expression pattern of this gene within the fly brain. And we've worked very closely with Larry Zapersky's lab to build a series of reporter genes where we've set this up. It's rather complicated, but the idea is that when the exon that you're interested in looking at is included, you express the GAL4 transcription factor, which then can in turn activate expression of a GFP reporter gene. And so we have one fly for each reporter for each of these exons. And what this allows us to do is basically look at the expression pattern of each exon at single neuron resolution in the fly. And this is just an example of 4 of the 12 different exons that we've looked at. So 4.1, 4.2, 4.9, and 4.12. And this is the mushroom body of the fly, which is very similar to the hippocampus of humans. And so what you can see is this exon 4.1 is expressed in sort of a moderate number of neurons. 4.2 is expressed in very, very few of them. And 4.12 is expressed in a lot of these. So this is really nice, but there's really too many neurons here to do anything quantitative with this. So there's another system that we can look at in the fly, which are these things called DA neurons, which are on the peripheral of the fly larvae. So this is a fly larvae from head to tail, and if you segment it down the middle and sort of fillet it open, there's 7 rows of neurons and 3 columns in each side. And so in every single animal, you can pinpoint the identical neuron between the animals. So you can ask, how many or how often is that neuron expressing a particular exon between individual flies. And so we can quantitate this, and basically the bottom line of this is that, for instance, the blue is on and the red is off, but the bottom line is that, for instance, exon 4.2 is expressed in almost all of the neurons, in these DA neurons, and exon 4.1 is expressed in very few, but it's expressed in different neurons in every individual animal, but it's almost always the same number of neurons. So what we think this means is that the splicing of this gene occurs in sort of a probabilistic manner, and that is, for instance, if we look at mushroom body neurons for exon 4.1, it's expressed in a moderate number there, but very few neurons in the DA neurons, whereas exon 4.2 is sort of the opposite. So we think it's the probability of splicing, and we think that the RNA binding proteins that are expressed in these neurons probably determine the expression level of these, and we think it's sort of like a 12-sided dice. So another really interesting phenomenon that we've been studying in Drosophila is this process of trans-splicing. And this is a case where instead of making a single pre-messenger RNA and putting the exons together, you actually make multiple RNAs and they get spliced together in trans. So this is the first gene that was known to be trans-spliced, which is the modifier of MDG4 gene. And it has these 4 common exons that are spliced to one of these purple exons. And the interesting thing is that several of these exons are on the opposite strand of RNA from the common exons. So the only way that they can be spliced together is to make 2 different RNAs and then splice them together in trans. And then this lola gene in Drosophila has a very similar organization, except there's no backwards exons. So this gene was known to be discovered to be trans-spliced through a series of complementation experiments that I won't get into. But we've been working on trying to figure out the mechanisms of this for a few years now. And some of the findings that I'll just summarize here are that for the mod MDG4 gene, you make these different precursor transcripts. And we think that these are then co-transcriptionally spliced together. So we think both the common and the variable exons, the splicing of them together occurs co-transcriptionally. And that you can have this occurring between exons that reside on the same allele and actually across alleles, so from one allele to the other. And I'll get into more detail on that in a minute. And then for the lola gene, we see the same thing, except there's only a single transcript expressed from each gene, so you don't have multiple promoters. And so we think trans-splicing occurs sort of by these transcripts from different alleles getting entangled with one another. And so one way we can look at this is if we take 2 different Drosophila species that are highly inbred, so that we know genomes are completely phased, and we can mate these together to generate F1 hybrids and collect RNA at various times. And then we can perform a sequencing assay, which I won't get into, but we can tell whether the 2 exons that are spliced together come from the same allele or the opposite allele. And what we see is that we can see splicing of exons from the same allele occur early, and then there's a lag. But for both mod and lola, we see a lag where we see splicing between the 2 alleles occurs after splicing of the exons between the same allele. And what we actually think this means and how it occurs is that we think the trans-splicing requires chromosomal pairing. Because if we do FISH to look at where the chromosomes are within the cell, prior to the onset of trans-splicing, we see the alleles are separate from one another within the cell. And then immediately at the time of observing trans-splicing and after, we see that these chromosomes are paired after. So we only see one locus per gene in these experiments. So I think chromosome pairing brings these two alleles together, and that allows the RNAs to get trans-spliced to one another. Okay, so that's Drosophila, but we're here about personalized medicine and human genomics, really. So I've been very pleased to hear that almost all of the speakers today have really helped me out, and Bob will probably do this more later, in showing that RNA binding proteins and mutations that have an impact on some aspect of RNA biology play roles in various human diseases. And there's a lot of RNA binding proteins in the genome, and for the vast majority of these things, we don't know what they do. And even the ones that we do know about them, we don't really know how they work very well. So this is just a schematic. We think there's approximately 1,000 RNA binding proteins in the human genome, and this is a schematic. And as I said, we don't know what the vast majority of these things do. So my lab is heading up a component of the ENCODE project right now in human. And our goal is to really comprehensively characterize the role of about 250 RNA binding proteins in the human genome. And we're doing this using a multi-pronged approach. So one of them is that we're using this approach called ClipSeq, or alternatively HITS-CLIP, which actually Bob Darnell developed and pioneered, which is similar to the ChIP-seq type experiment for transcription factors or chromatin factors, and allows you to identify the binding sites for each protein with the RNA at very high resolution. So that for 250 of these proteins, we're going to be identifying the binding sites throughout the transcriptome for them. We're also going to be doing experiments where we knock that protein down and then analyze what happens to the transcriptome so that we can try and identify the functions and assign functions for the binding sites for these proteins. And then another type of experiment that Chris Burge's lab is doing with us is this thing called bind and seq, which is basically an experiment that allows you to determine the affinity of each RNA binding protein for all possible sequences. And where this will be really critical is in interpreting human variation. So as we find mutations within the human genome that overlap with a binding site for one of these RNA binding proteins, with using this particular data, we'll be able to predict whether or not that mutation would impact the binding of that protein or not, and if so, how much. So this project's about 2 years underway, and we've been really generating lots of reagents. We've screened almost 1,000 antibodies for doing ClipSeq and lots of shRNAs, and the data is now finally being produced. And this is the ENCODE project is a community resource project. The idea is to generate data that everybody will use. And so you can go to this website, and the data is just coming out, and it's all freely available. And I'd appreciate it if you don't scoop us, but anyway, so this is the group that is working together with us on the ENCODE project in California. So Jean Yo's lab is doing the ClipSeq, Fu is doing some ChIP-seq, Chris Burge is doing the bind and seq, and one thing I didn't say, Eric Lecoyer from McGill, he's actually doing protein localization for all of these proteins as well. And then finally, the rest of my lab is shown here, and they basically do everything. And then the ModENCODE collaborators are here, and I have to thank all of these people for funding, and thank you for your attention, and I have to answer questions, or you can just go to lunch. So.